Introduction.
Hey Ankita, we know that the cell is the structural and functional unit of life, but do you know what does it actually mean? Yeah, it means a single cell can perform all basic functions of life. Example, movement, intake of food and respiratory gases, respiration and excretion. Yes, you're right. So, come let's start our new chapter, Cell the Unit of Life.
Objectives. At the end of this lesson, you will be able to define the cell, understand the cell theory, know the characters of the cell, understand functional regions of a cell, know the different shapes of the cell, understand different cell sizes. The cell. Cells are the structural and functional unit of all living organisms and are building blocks of the human body. Adult human body contains 75 trillion cells. Each cell type performs specific function. There are 200 cell types in humans. The term cell was first coined by an English microscopist, Robert Hooke, in 1665. He discovered cells while looking in a thin slice of cork and called tiny boxes of a honeycomb as the cells. He thought that cells only exist in plants and fungi. Antony van Leeuwenhoek was the first to observe free cells like bacteria, protozoa, red blood cells and sperms, etc. using his homemade microscope. He called them animalcules. He also observed blood cells from fish, birds, frogs, dogs and humans. Therefore, it was known that cells are found in animals as well as plants. The cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that plants are composed of different kinds of cells which form the tissues of the plant. In 1839, Theodore Schwann, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. He concluded that the presence of cell wall is a unique character of the plant cells. In 1839, Schleiden and Schwann together formulated the cell theory. In 1858, Rudolf Virchow, a German physician, after extensive study of cellular pathology, concluded that cells must arise from pre-existing cells. Modern cell theory contains 4 statements in addition to the original cell theory. Characteristics of cells. Cells are highly complex and organized. They possess a genetic program and the means to use it. They are also capable of producing. They acquire and utilize energy. Even they carry out a variety of chemical reactions and mechanical activities. They are also able to respond to stimuli and self-regulation. Functional regions of cells. There are 3 functional regions of cell: plasma membrane, cytoplasm, and nucleus. Plasma membrane. Plasma membrane is the outermost thin, transparent, elastic, and semi-permeable membrane. Cytoplasm. Cytoplasm is an amorphous and homogeneous colloidal substance present between the plasma membrane and nucleus. Nucleus. Nucleus is a membrane-bound structure which contains DNA in its chromosomes. Cell shape. There are different shapes of cells in various organisms. In multicellular organisms, there are different shapes of cells in different parts of body. Amoeba and leukocytes do not have a fixed shape as they have pseudopodia. Cheek cells are flat. Germ cells are cuboidal. Intestinal cells are columnar. Liver cells are polygonal. Fat cells are rounded. Paramecium is slipper-shaped. Hence, egg is oval. Smooth muscles are spindle-shaped. Pigment cells, that is, chromatophores, are branched. Muscles or nerve fibers are long thread-like. Guard cells of stomata of dicots are bean-shaped, while of monocots are dumbbell-shaped. Cell sizes. The size of cells varies greatly in different organisms. The smallest known cells are of pleuropneumonia-like organism, that is PPLO, Mycoplasma gallisepticum, of about 0.1 micrometer size. Largest animal cell is of ostrich egg and measures about 170 by 135 millimeter. Prokaryotic cell size ranges from 1 to 10 micrometer, while eukaryotic cell size ranges from 10 to 100 micrometer. In plants, largest cell is of Acetabularia, green algae, which measures about 6 to 10 centimeter. In human body, most of the cell range between 2 to 3 micrometer, 20 micrometer and 30 micrometer in diameter. Nerve cells is the longest cell in human body, about 3 feet long. Did you know the term cytology was coined by Hertwig in 1893? Max Schultz in 1861 proposed protoplasm theory. Sachs proposed organismal theory in 1874. Summary. Let us summarize what we have learnt. Cells are broadly divided in two types, plant cells and animal cells. Lamarck for the first time stated that living beings are made up of cells. The term protoplasm was coined by Purkinje. Cell theory was put forward by Schleiden and Schwann in 1838 to 1839. They were German biologists. An exception to cell theory is virus. The term cell was coined and was first seen by Robert Hooke. The cells discovered in thin sections of cork by Robert Hooke were actually cell walls. Nucleus was discovered by Robert Brown. Virchow proposed cell lineage. Cell always arises from pre-existing cell. He said, "Hominis cellulae cellula." The term sarcode was used for living substance of cell by Jordan. Minimum cell size seen under light microscope is 0.25 micrometer. Protoplasm is complex colloidal solution and forms 95% of total weight of the body. It is called physical basis of life. An individual has a number of different types of cells was first stated by Dutrochet. The cells vary in their shape, size, and activities or functions. In unicellular organisms, the cell shape is maintained by tough plasma membrane, example, paramecium and exoskeleton. Fixed shape of the cell occurs in most plants and animals. In multicellular organisms, the cell shape depends on its functional adaptations and partly on the surface tension, viscosity of the protoplasm, the mechanical action exerted by adjoining cells, and rigidity of the cell membrane. Example, presence of rigid cell wall in plant cells. Thus, cell may have diverse shapes such as polyhedral with 8, 12 or 14 sides, spherical, example, eggs of many animals, spindle-shaped, example, smooth muscle fibers, elongated, example, nerve cells, branched, example, chromatophores or pigment cells of skin, discoidal, example, erythrocytes or red blood cells, etc. The size of different cells ranges between broad limits. Some plants and animal cells are visible to the naked eye. Most cells are visible only with the microscope since they are only a few micrometer in diameter. A micrometer is 1/1000th of a millimeter. The size of cells varies from the very small cells of bacteria, 0.2 to 5 micrometer, to the very large eggs of the ostrich, 18 centimeter, and among plants, the largest cell is the ovule of cycas. Some nerve cells of human beings have a meter-long tail or axon. Single marine algae, Acetabularia, measures nearly 10 centimeter in height. Prokaryotic cells usually range between 1 to 10 micrometer. The eukaryotic cells are typically larger, mostly ranging between 10 to 100 micrometer. The size of unicellular organisms is larger than a typical cell of a multicellular organism. Example, amoeba protus is biggest among the unicellular organisms, its length being 0.6 millimeter, that is 60 micrometer. The size of typical cells of multicellular organisms ranges between 20 to 30 micrometer. The smallest cells are those of Mycoplasma gallisepticum, an organism intermediate between virus and bacteria. Their size is about 0.1 micrometer. Introduction.
Ankita, we have discussed cell theory, structure and functional regions of cell already. Yes, and now we'll discuss that there are two types of cells, plant cells and animal cells. Okay, we'll also observe structure of plant and animal cells and also discuss difference between plant and animal cells, that is prokaryotic and eukaryotic cells.
Objectives. At the end of this lesson, you will be able to identify types of cells, know the characters of plant cells and animal cells, understand difference between plant and animal cells, know the characters of prokaryotic cell, understand cell envelope of prokaryotic cell, know the characters of ribosome and inclusion bodies of prokaryotic cell, understand difference between prokaryotic and eukaryotic cells. Types of cell. We have two types of cells, plant cell and animal cell. On the basis of presence of nuclear membrane, there are two types of cells, prokaryotic and eukaryotic. Characteristics of plant cells. All the living matter of a plant cell is known as protoplasm. The cell is surrounded by a cell membrane or plasma membrane. Plant cells also have a cell wall surrounding plasma membrane. Cell wall is made up of cellulose and is rigid, so it supports the plant cell. The nucleus is the control center of the cell and is surrounded by cytoplasm. The vacuole is a storage area for the plant cell which contains cell sap made up of sugars, salts and pigments. Plant cells also have chloroplasts which contain chlorophyll. It is the place where photosynthesis occurs within the cell. Characteristics of animal cells. All the living matter of an animal cell is known as protoplasm. The cell is surrounded by a cell membrane or plasma membrane. The nucleus is the control center of the cell and is surrounded by cytoplasm. Cytoplasm is composed of 90% water. There are many organelles within the cytoplasm. In cytoplasm, most of the cell's activities take place. Difference between plant and animal cells. In plant cell, the cell wall is present outside the plasma membrane, while in animal cell, the cell wall is absent. Shape of cell is firm in plant cells, while in animal cells, due to absence of cell wall, cell shape is not fixed. In plant cell, protoplasm is peripheral, while in animal cell, it fills whole cell. In plant cells, nucleus lies in peripheral protoplasm and is oval in shape, while in animal cell, nucleus is central in position and is round in shape. In plant cells, large vacuoles lie in the center with cell sap, while in animal cells, vacuoles, if present, are few and small. Chloroplasts are usually present in the plant cells, while in animal cells, chloroplasts are usually absent. Characters of prokaryotic cells. The prokaryotic cells are primitive. Bacteria and blue-green algae, that is, cyanobacteria, have such types of cells. Fully developed cell organelles are not present in such cells. In the prokaryotic cells, nucleus is without a nuclear membrane, due to which substances usually present in the nucleus are diffused in the central part of the cell in direct contact with the cytoplasm. Characters of cell envelope and its modification. Prokaryotic cells have a chemically complex cell envelope, which is made up of three-layered structure. The outer layer of glycocalyx, middle layer is cell wall, and innermost layer is plasma membrane. Glycocalyx is either in the form of loose sheath called slime layer, or it is thick and tough layer which is called capsule. The cell wall gives a strong structural support to the cell, which prevents the cell to burst or collapse. It also provides shape to the cell. The plasma membrane is semi-permeable as it is in eukaryotes. According to the Gram staining method invented by Hans Christian Gram, the bacteria can be divided into two types. In Gram-positive bacteria, peptidoglycans of cell envelope takes up the crystal violet stain. In Gram-negative bacteria, peptidoglycans of cell envelope do not take up the crystal violet stain. Mesosomes of prokaryotic cells. In prokaryotic cells, a special membranous structure, mesosome, is found. It is formed by extensions of plasma membrane into the cell. The mesosomes help in respiration and secretion processes. The mesosomes also help to increase the surface of plasma membrane. As a result, increase the enzymatic content of cell. The mesosomes also help in formation of cell wall, DNA replication and distribution to daughter cells. Characters of ribosome of prokaryotic cells. In prokaryotes, ribosomes are about 15 nanometer by 20 nanometer in size and are associated with the plasma membrane of the cell. Ribosomes are made up of two subunits, 50S and 30S units. They combine to form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Many ribosomes attach to single mRNA and form a chain known as polyribosome or polysome. The ribosomes of a polysome translate mRNA into proteins. Movement of prokaryotic cells. Prokaryotes are motile or non-motile. When motile, they have flagella outside their cell wall. These flagella are thin and filamentous. Flagella are made up of three parts: filament, hook, and basal body. Filament is the longest portion of flagella, which extends from the cell surface to the outside. Flagella may be one or many. Pili and fimbriae are also present on the surface of prokaryotic cells. Pili are made up of special protein and are elongated tubular structures. The fimbriae are small bristle-like fibers coming out of the cell. Characters of inclusion bodies of prokaryotic cells. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies, which lie free in the cytoplasm. Example: phosphate granules, cyanophyceae granules, and glycogen granules. Difference between prokaryotic and eukaryotic cells. Prokaryotic cells are smaller, less than 5 micrometer, while eukaryotic cells are larger, greater than 10 micrometer. Prokaryotic cells are always unicellular, while eukaryotic cells are mostly multicellular. Prokaryotic cells do not have membrane-bound nucleus and organelles, while eukaryotic cells have membrane-bound nucleus and organelles. In prokaryotic cells, DNA is circular and without proteins, while in eukaryotic cells, DNA is linear and is associated with proteins to form chromatin. In prokaryotic cells, ribosomes are small 70S, while in eukaryotic cells, ribosomes are larger 80S. Prokaryotic cells do not have cytoskeleton, while eukaryotic cells always have a cytoskeleton. Prokaryotic cells move by rigid rotating flagellum, while eukaryotic cells move by waving cilia or flagella. Prokaryotic cells divide by binary fission, while eukaryotic cells divide by mitosis or meiosis.
Did you know?
Nucleoid is also known as genophore. When plasmid associates with nucleoid of bacterium, then it is called episome. In mycoplasma, cell wall is absent. Summary. Let us summarize what we have learned. There are two types of cells, plant cells and animal cells. Plant cells have a cell wall outside the cell membrane. Based on the presence or absence of a membrane-bound nucleus and other organelles, cells and hence organisms can be named as eukaryotic or prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus and cytoplasm. Bacteria are ubiquitous in every habitat on Earth. Prokaryotic cells usually range between 1 to 10 micrometer. The eukaryotic cells are typically larger, mostly ranging between 10 to 100 micrometer. The prokaryotic cells are primitive. Fully developed cell organelles are not present in such cells. In the prokaryotic cells, nucleus is without a nuclear membrane, due to which substances usually present in the nucleus are diffused in the central part of the cell in direct contact with the cytoplasm. Bacteria and blue-green algae, that is, cyanobacteria, have such types of cells. Introduction.
We have discussed cell theory, structure and functional regions of cell, types of plant and animal cells, structure of plant and animal cells, prokaryotic cells, and difference between plant and animal cells already. Yes, now we'll discuss structure of eukaryotic cells and also observe the structure and functions of cell membrane and cell wall.
Objectives. At the end of this lesson, you will be able to know the characters of eukaryotic cell, know the characters of cell membrane, understand the functions of cell membrane, know the characters of cell wall, understand the functions of cell wall. Structure of eukaryotic cells. Eukaryotic cells have a definitely organized nucleus and cell organelles. These types of cells are found in many groups of algae, except cyanobacteria, fungi, and other plants and animals. A typical eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Genetic material is organized in the form of chromosomes in eukaryotic cells. Eukaryotic cells also have cytoskeletal structures. Characteristics of cell membrane. The cell membrane is also called the plasma membrane or plasmalemma. It is one biological membrane separating the interior of a cell, that is, a living cell, from the outside environment, that is, non-living surroundings. The cell membrane consists of both lipids and proteins along with carbohydrates. The most abundant lipids are phospholipids. The membranes are assemblies of lipid and protein molecules held together by non-covalent interactions. The lipid molecules are arranged as a continuous bilayer, that is, double layer, approximately 4 to 5 mm thick. Fluid mosaic model. The arrangement of phospholipids and proteins in biological membranes is described by the fluid mosaic model presented by S. J. Singer and G. Nicholson in 1972. The biological membranes can be considered as a two-dimensional liquid where all lipid and protein molecules diffuse more or less easily. The cell membrane consists primarily of thin layer of amphipathic molecules having both hydrophobic water-hating region and hydrophilic water-loving regions, phospholipids, which spontaneously arrange so that the hydrophobic tail regions are shielded from the surrounding polar fluid, causing the more hydrophilic head regions to associate with the cytosolic and extracellular phases of the resulting bilayer. The arrangement of hydrophilic heads and hydrophobic tails of the lipid bilayer prevent polar solutes, example, amino acids, nucleic acids, proteins, carbohydrates, and ions from diffusing across the membrane, but generally allows for the passive diffusion of hydrophobic molecules. Associated with each membrane is a set of membrane proteins that enables the membrane to carry out its distinctive activities. Membrane proteins can be classified in two broad categories: integral, that is intrinsic; peripheral, that is extrinsic. Integral membrane proteins have one or more segments that are embedded in phospholipid bilayer, having one or more covalently attached fatty acid chains that help them anchor to the membrane. Peripheral membrane proteins do not interact with the hydrophobic core of the phospholipid bilayer. Instead, they are usually bound to the membrane indirectly by interactions with integral membrane proteins. These proteins interact with membranes in different ways on the cytoplasmic side of the membrane. Some membrane protein connect to the cytoskeleton. On the exterior side of the membrane, some membrane proteins attach to the fibers of the extracellular matrix. Functions of cell membrane. Holding cell contents. Cell membrane hold the semi-fluid protoplasmic contents. Individuality. Plasmalemma maintains form and individuality of the cell. Protection. Plasma membrane protects the animal cells from injury. Plant cells possesses wall over the plasmalemma for extra protection and support. Organelles. Cell membranes delimit a number of subcellular structures or organelles like nucleus, mitochondria, plastids, Golgi apparatus, endoplasmic reticulum, microbodies, etc. Cell recognition. With the help of glycolipids and glycoproteins, cell membranes is able to differentiate similar cells from dissimilar ones, foreign substances and tissues from familial materials. Cell recognition is useful for tissue formation and defense against microbes. Antigens. Cell membrane possesses antigens which determine blood grouping, immune response, acceptance or rejection of a transplant. Cell junctions. They help in keeping animal cells together. Sheaths. Plasma membrane forms sheaths around cilia and flagella. Microvilli. These are membrane evaginations which are specialized for increasing absorption. Cytoplasmic bridges. Plasmodesmata and gap junctions form cytoplasmic bridges between adjacent cells. Osmosis. It is caused by the presence of special water channels in the membranes. Endocytosis. Bulk intake of materials or endocytosis occurs through development of membrane vesicles. Exocytosis. It is reverse of endocytosis that provides for throwing out of waste products and secretory materials. Intercellular communication. Plasma membrane establishes a flexible boundary, protects cellular contents, and supports cell structure. Physical barrier. Phospholipid bilayer separates substances inside and outside the cell. Selective permeability. It regulates entry and exit of ions, nutrients, and waste molecules through the membrane. Structure of cell wall. A cell wall is a tough, usually flexible, but sometimes fairly rigid layer that surrounds some types of cells. It is located outside the cell membrane and provides these cells with structural support and protection, and also acts as a filtering mechanism. They are found in plants, bacteria, fungi, algae, and some archaea, animals, and protozoa. The chief structural component of cell wall is cellulose, which is the chain of many thousand sugar units. Along with cellulose, pectic compounds, hemicellulose, lignin, suberin, chitin, protein, fats, etc. may also be found in the cell wall. Key nutrients, especially water and carbon dioxide, are distributed throughout the plant from cell wall in apoplastic flow. Middle lamella. The plant cell wall can be differentiated into 4 regions: middle lamella, primary wall, secondary wall, tertiary wall. The outermost layer of cell wall, or the intercellular structure separating the walls of 2 adjacent plant cells, is middle lamella. The pectins are most commonly found in the middle lamella. The basic chemical unit of pectin is the carbohydrate galacturonic acid, which is capable of forming salts with calcium and magnesium. The middle lamella is mainly made up of calcium pectate. The characteristic softening of fruits during ripening is mainly due to increase in the solubility of middle lamella. Primary wall. The initial wall of a plant is the primary wall. It is found inner to middle lamella. The primary wall develops on middle lamella on both the sides. The primary wall is composed of cellulose, hemicellulose, pectins, and lignin. The primary cell wall of most plant cells is semi-permeable and permits the passage of small molecules and small proteins, with size exclusion estimated to be 30 to 60 kDa or 30,000 to 60,000 atomic mass. Secondary wall. The primary wall is followed by secondary wall, which is thick, rigid, permeable, and lies near the plasma membrane, or tertiary cell wall, mainly composed of cellulose, hemicellulose, xylin, lignin, etc. In some cells, the secondary wall is impregnated with the lignin to form wood. Lignin is coniferal, complex polymer of aromatic compound and is the main constituent of woody tissues. In some cells, the secondary wall is impregnated with suberin to form cork, making it impervious to water. Tertiary wall. The tertiary wall is present beneath the secondary wall in tracheids, the principal water-conducting cells of gymnosperms. The tertiary wall is special xylin-rich wall layer deposited inner to the secondary wall in tension wood of some gymnosperms. Cellulose microfibrils are absent. Tertiary wall is distinguishable both chemically as well as through staining. Functions of cell wall. A major function of the cell wall is to act as pressure vessel, preventing overexpansion when water enters the cell. Selective permeability regulates entry and exit of ions, nutrients, and waste molecules through the membrane. It determines the shape of a plant cell and prevents desiccation of cells. Plasmodesmata connections help in movement of substances between cells. Cells connected by plasmodesmata form a system called symplast. The cell wall permits the plant cell to become turgid. As water enters the vacuole by osmosis, the plant cell expands. The cell wall has to be strong enough to resist this expansion and so enable the cell to become turgid. Apposition is the addition of new cell wall particles amongst the existing ones. It provides mechanical strength to support the cell. The cellulose microfibrils are very strong. The strength may be increased by the addition of lignin in tissues such as xylem. In cells such as collenchymus, extracellulose is added to the cell wall to increase mechanical support. The cell wall has narrow pores called pits, through which fine strands of cytoplasm called plasmodesmata are able to pass. These intercellular connections allow exchange of materials between the living cell contents.
Did you know?
Nageli and Kramer introduced the term cell membrane in 1855. In 1931, J.Q. Plowey called cell membrane as plasma lemma. Danieli and Davison proposed sandwich model of plasma membrane in 1935. According to this model, it is a protein-lipid bilayer protein sandwich. In 1959, J. David Robertson called all the biomembranes as unit membranes. Summary. Let us summarize what we have learned. Plasma membrane is not permeable to glucose 6-phosphate. Membrane carbohydrates are usually branched oligosaccharides with fewer than 15 sugar units. They may be covalently bonded to lipids forming glycolipids or more commonly to proteins forming glycoproteins. The plasma membrane is selectively permeable and facilitates transport of several molecules. The plasma membrane consists of both lipids and proteins along with carbohydrates. The cell membrane consists primarily of thin layer of amphipathic phospholipids which spontaneously arrange so that the hydrophobic tail regions are shielded from the surrounding polar fluid, causing the more hydrophilic head regions to associate with the cytosolic and extracellular phases of the resulting bilayer. The latest model of plasma membrane is fluid mosaic model. It was put forward by Singer and Nicholson. According to the fluid mosaic model, plasma membrane is composed of phospholipids, extrinsic proteins and intrinsic proteins. Proline is not constituent of cell membrane. The plasma membrane helps in osmoregulation. Plasma lemma is made of single lipid layer and 2 protein layers. Correct sequence of protein P and lipid L in cell membrane is PLLP. Lipid molecules of plasma membrane are arranged parallel. Cell recognition and adhesion occur due to biochemicals of cell membranes named glycoproteins and glycolipids. 2 animal cells are interconnected by desmosome. Cell eating is phagocytosis. The plasma membrane contains receptors that recognize and respond to molecular signals. The plasma membrane establishes a flexible boundary, protects cellular contents, and supports cell structure. Phospholipid bilayer separates substances inside and outside the cell.
Introduction. We have discussed structure of eukaryotic cells and structure and functions of cell membrane and cell wall already. Yes, now we will discuss structure of endomembrane systems. Okay, we will also observe structure of endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, mitochondria, plastids, and ribosomes.
Objectives. At the end of this lesson, you'll be able to find the characters of endomembrane systems, find the characters of endoplasmic reticulum, understand characters of Golgi apparatus, find the characters of lysosomes, find the characters of vacuoles, find the characters of mitochondria, understand characters of plastids, find the characters of ribosomes. Characters of endomembrane system. A system of interconnected membranes, also known as the cytomembrane system, that fills the cell interior and connects the cell boundary with the double-membraned organelles, nucleus, plastids, and mitochondria. The phospholipid bilayer forms the basis for both outer and internal membranes. Thus, while the plasma membrane, the outermost cell membrane, is an integral component of the cell boundary, its numerous convoluted protrusions into the cell interior and its connectivity with the cytomembranes also allow it to be considered a part of the endomembrane system. The endomembrane system organizes the eukaryotic cell into numerous specialized functional domains that are recognized as different organelles. Major endomembrane-derived organelles of the plant cell are endoplasmic reticulum, Golgi bodies, peroxisomes, vacuoles, endosomes, autophagosomes, outer nuclear envelope. Structure of the endoplasmic reticulum. The endoplasmic reticulum ER is an organelle of cells in eukaryotic organisms that forms an interconnected network of tubules, vesicles, and cisternae. The lacy membranes of the endoplasmic reticulum were first seen by Keith R. Porter, Albert Claude, and Ernest F. Fulham in 1945. There are two regions of the ER that differ in both structure and function. One region is called rough ER because it has ribosomes attached to the cytoplasmic side of the membrane. The rough ER is a series of flattened sacs. Rough endoplasmic reticula synthesize proteins. The other region is called smooth ER because it lacks attached ribosomes. The smooth ER is a tubule network. Smooth endoplasmic reticula synthesize lipids, steroids, and morphine, metabolize carbohydrates and steroids, and regulate calcium concentration, drug metabolism, and attachment of receptors on cell membrane proteins. The space inside of the ER is called the lumen. The ER is very extensive, extending from the cell membrane through the cytoplasm and forming a continuous connection with the nuclear envelope. Characters of Golgi apparatus. Golgi apparatus is a membranous organelle composed of many flattened sacs. The Golgi apparatus forms a part of the cellular endomembrane system. It was identified in 1898 by the Italian physician Camillo Golgi, after whom the Golgi apparatus is named. The secretions of cells are packed in them and transported from the cell. Golgi apparatus consists of a set of membrane-bound cisternae, vesicles, and secretory vesicles having polarity. It processes and packages proteins inside the cell, and before they make their way to their destination, it is particularly important in the processing of proteins for secretion. Characters of lysosomes. Lysosomes were discovered by the Belgian cytologist Christian D. Dove in the 1960s. The name lysosomes derives from the Greek words lysis, to separate, and soma, body. They are frequently nicknamed suicide bags or suicide sacs by cell biologists due to their autolysis. Lysosomes are cellular organelles that contain acid hydrolase enzymes to break down waste materials and cellular debris. They can be described as the stomach of the cell. They are found in animal cells, while in yeast and plants, the same roles are performed by lytic vacuoles. Lysosomes digest excess or worn-out organelles, food particles, and engulf viruses and bacteria. The membrane around lysosomes allows the digestive enzymes to work at the 4.5 pH they require. They are created by the addition of hydrolytic enzymes to early endosomes from the Golgi apparatus. Lysosomes fuse with vacuoles and dispense their enzymes into the vacuoles, digesting their contents. Characters of vacuoles. A vacuole is a membrane-bound organelle which is present in all plant and fungal cells and some protist, animal, and bacterial cells. Vacuoles are essentially enclosed compartments which are filled with water containing inorganic and organic molecules, including enzymes in solution, though in certain cases they may contain solids which have been engulfed. Vacuoles are formed by the fusion of multiple membrane vesicles and are effectively just larger forms of these. They have no basic shape or size. Its structure varies according to the needs of the cell. The functions of the vacuole includes isolating materials that might be harmful or a threat to the cell, containing waste products, containing water in plant cells, maintaining internal hydrostatic pressure or turgor within the cell, maintaining an acidic internal pH, containing small molecules, exporting unwanted substances from the cell, allows plants to support structures such as leaves and flowers due to the pressure of the central vacuole. In seeds, stored proteins needed for germination are kept in protein bodies which are modified vacuoles. Characters of mitochondria. Mitochondria are membrane-bound organelles and, like the nucleus, have a double membrane. They are about the size of bacteria but may have different shapes depending on the cell type. The outer membrane is fairly smooth, but the inner membrane is highly convoluted, forming folds called cristae. The cristae greatly increase the inner membrane's surface area. It is on these cristae that food, sugar is combined with oxygen to produce ATP, the primary energy source for the cell. Mitochondria has its own DNA and ribosomes. Mitochondria provide the energy a cell needs to move, divide, produce secretory products, contract. In short, they are the power centers of the cell. Characters of ribosomes. George Palade in 1953 under electron microscope observed dense particles. Ribosomes are granular structure. Ribosomes are made of ribonucleic acid RNA and proteins. They are not membrane bound. The eukaryotic ribosomes are 80S and prokaryotic ribosomes are 70S. S is Swedberg unit and stands for the sedimentation coefficient. It is a measure of density and size. Both 80S and 70S ribosomes are made up of two subunits. Ribosomes are also attached to the outer membrane of endoplasmic reticulum. Ribosomes take part in protein synthesis. Characters of plastids. Plastids are found in all plant cells and also in euglenoids. They are large in size, so they can be easily observed under the microscope. They have some specific pigments, so they give color to the plants. On the basis of pigments present in plastids, they can be of three types: chloroplasts, chromoplasts, and leucoplasts. The chloroplasts have chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. Chromoplasts have fat-soluble carotenoid pigments like keratin, xanthophylls. They give yellow, orange, or red color to the plants. Leucoplasts are colorless plastids of various shapes and sizes. They are amyloplasts which store carbohydrates, example, potato, elaioplasts which store oils and fats, and aleuroplasts which store protein. Characters of chloroplasts. Chloroplasts are found in mesophyll cells of leaves. They are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles ranging from 5 to 10 micrometer long and 2 to 4 micrometer wide. There may be one chloroplast, as in Chlamydomonas, or 20 to 40 per cell, as in green algae. Chloroplasts are double membrane bound. The inner membrane is less permeable. The space limited by inner membrane is known as stroma. There are a number of organized flattened sacs called thylakoids in stroma. These thylakoids are arranged in stacks like the piles of coins and are known as grana. The thylakoids of different grana are connected by the flat membranous tubules called stroma lamella. The space enclosed by membrane of thylakoids is known as lumen. The stroma contains carbohydrates, proteins, double-stranded circular DNA, and ribosome 70S. Thylakoids contains chlorophyll pigments as well. Did you know? Lysosomes were earlier known as pericanalicular dense bodies. Main enzyme present in Golgi apparatus membrane is nucleoside diphosphate. Liver cells have both smooth and rough endoplasmic reticulum. Mitochondria and chloroplasts are self-duplicating cell organelles. In giant amoeba chaos, there are about 50,000 mitochondria. Summary. Let us summarize what we have learned. SER is mainly found in cells actively engaged in lipid metabolism. The smooth endoplasmic reticulum SER is detoxification site in liver. Organelles connected with lipid synthesis are SER. Granular endoplasmic reticulum differs from SER in having ribosomes on its surface. Gastric cells secreting zymogen have well-developed RER. RER is well developed in cells engaged in synthesis of secretory products. Function of RER is protein synthesis. RER differs from SER in presence of ribosomes. The Golgi complex occurs in all cells except erythrocytes and bacterial cells in perinuclear area. They are absent in bacteria and blue-green algae. The Golgi complex is a membranous organelle composed of flattened sacs. The secretions of cells are packed in them and transported from the cell. The Golgi complex is specialized for glyosidation of lipids and proteins. Golgi complex produces vesicles and lysosomes. ETC is component of Golgi complex. Golgisome is involved in transformation of cell membranes and protein packaging. The Golgi complex is exported house of the cell. Lysosomes were discovered by D. Dove. They are called so because they have carboxylating enzymes. Lysosomes are formed Golgi bodies. Lysosomes are also called suicidal bags because they have hydrolytic enzymes which function at acidic pH 5. Lysosomes are single membrane structures containing enzymes for digestion of all types of macromolecules. Double membrane or unit membrane is absent in lysosomes. Lysosomes are membranous bag with hydrolytic enzymes which is used for controlling intracellular digestion of macromolecules. Lysosomes are polymorphic and contain hydrolases. Lysosomes reduce the number of other organelles. Autolysis is connected with lysosomes. If lysosomes get ruptured in a cell, it dies. Function of lysosomes is breakdown of cell substances. When lysosomes fuses with phagosome, it results in the formation of secondary lysosomes. Cell vacuoles contains water and dissolved substances. Tonoplast, a differentially permeable membrane, surrounds vacuole. Vacuoles are of 4 types: contractile vacuoles, sap vacuoles, food vacuoles, and gas vacuoles. Vacuoles store food and waste. Some vacuoles store extra water. Cell sap occurs in vacuole. Function of vacuole is storage, waste disposal, and cell elongation and protection.
Introduction.
Hey Ankita, we have discussed structure of endomembrane system, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, mitochondria, plastids, and ribosomes. I think we have completed all the topics. No, Madhuri, there are some more topics to be learned. They are structure of cytoskeleton, cilia and flagella, centrosomes and centrioles, nucleus and microbodies. So come, let's know about them in this module.
Objectives. At the end of this lesson, you will be able to know characters of cytoskeleton, know the characters of cilia and flagella, understand characters of centrosomes and centrioles, know the characters of nucleus, know characters of microbodies. Characters of cytoskeleton. Cytoskeleton is an elaborate network of filamentous, proteinaceous structures present in the cytoplasm. Cytoskeleton provides mechanical support, motility, maintenance of the shape of the cell. Characters of cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The cilia of flagella are covered with plasma membrane. Their core is known as exoneme. Exoneme possesses a number of microtubules running parallel to the long axis. The exoneme has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. This arrangement is known as 9+2 array. The central tubules are connected by bridges and are enclosed by a central sheath which is connected to one of the tubules of each peripheral doublets by a radial spoke. The peripheral doublets are also interconnected by linkers. Both cilia and flagella emerge from centriole-like structure called the basal bodies. Characters of centrosomes and centrioles. Centrosome is an organelle containing 2 cylindrical structure called centrioles. Centrosomes are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which Each has an organization like the cartwheel. Centrosomes are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is triplet. The central part of the proximal region of the centriole is also proteinaceous and called the hub. This hub is connected with tubules of the peripheral triplet by radial spokes made of protein. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to the spindle apparatus during cell division in animal cells. Characters of nucleus. Nucleus was at first discovered by Robert Brown in 1831 AD in orchid cells. Nucleus is also called the director of the cell. Nucleus is most important part of the cell, which directs and controls all the cellular functions of the body. Fleming named chromatin the material of the nucleus stained by basic dyes. The interphase nucleus has highly extended and elaborate nucleoprotein fiber called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Nuclear envelope has 2 parallel membranes with space between them known as perinuclear space. The outer membrane is usually continuous with the endoplasmic reticulum having ribosomes on it. Nuclear envelope is interrupted by minute spore at a number of places. These spores are formed by the fusion of its 2 membranes. The movement of RNA and protein takes place through these pores in both the directions between nucleus and the cytoplasm. Characters of chromosomes. During various stages of cell division, cells show structured chromosomes in the place of nucleus. Chromatin contains DNA and some basic proteins known as histones, some non-histone proteins, and RNA. A single cell of human being has about 2-meter-long DNA thread distributed among its 46 chromosomes. Each chromosome has a constriction point called the centromere, which divides the chromosome into 2 sections or arms. On the sides of the centromeres, disc-shaped structure called kinetochores are present. The short arm of the chromosome is labeled the P arm. The long arm of the chromosome is labeled the Q arm. Based on the position of the centromere, the chromosomes are classified into 4 types: metacentric, submetacentric, acrocentric, and telocentric. The metacentric chromosome has centromere in the middle, forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle so that one arm is shorter and one arm is longer. The acrocentric chromosome has centromere close to its end, forming one extremely short arm and one very long arm. The telocentric chromosome has terminal centromere. In some cases, the chromosomes have non-staining secondary constrictions at the constant location, which appears as a small fragment known as satellite. Special types of chromosomes. There are 2 special types of chromosomes: lamp brush chromosomes and polytene chromosomes. Lamp brush chromosomes occur inside oocytes of amphibian. Loops of lamp brush possesses DNA, RNA, and proteins. Lamp brush chromosomes are observed in meiotic prophase. Polytene chromosomes occur in cells of some insects. In oocytes, they occur in diplotin. Polytene chromosomes are formed due to duplication without separation and replication of DNA without cell division. Balbiani rings occur in polytene chromosome, which are sites of RNA and protein synthesis. Characters of microbodies. Many membrane-bound minute vesicles, known as microbodies that contain various enzymes, are present in both plant and animal cells. Microbodies include peroxisomes and glyoxisomes, which protect the cell from the damage of peroxides. Peroxisomes have glycolate oxidase enzyme, which helps in photorespiration. Spherosomes synthesize and store fat.
Did you know?
Nucleus was first observed by Leeuwenhoek in RBCs of fish. De Robertis et al. Gave ultrastructure of nucleus in 1971. In nucleus, DNA and histones ratio is nearly 1:1. Nucleolus is also called little nucleus. Nucleosomes are called functional units of chromosomes. Genophore is another name of nucleoid of prokaryotic cells. Summary. Let us summarize what we have learnt. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella are hair-like outgrowths of the cell membrane. Centrosome is an organelle usually containing two cylindrical structures called centrioles. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, cell is the structural and functional unit of life. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells.